Oh, hey Rose.
Good morning, sir.
Oh, you're super quiet. I mean, not in general, just your microphone.
Really?
Ah, that's a little bit better. Okay. I obviously wasn't just reading the news. Okay, so before we get started on this, uh, questions about anything else?
I had a question about insulators, and I
Huh?
wanted to know, do they only occur in between two genes to stop them from happening when they share an enhancer?
Yeah, so um, really they serve as bookends, right? Because you're going to have genes, kind of lined up on one strand and kind of going at the other direction as well. Um, if you do want to coordinate expression of two genes that are like so,
Mm-hmm.
then you would want the regulatory regions shared between those two to be able to affect both genes, right?
Oh, okay. Mm-hmm.
If you do want to coordinate expression between those two. However, if those are two completely different genes, let's say a uh, actin gene and I don't know, something involved in the immune system, right? So you really don't need those two things to be coordinated together. You want to basically isolate those genes from any other genes, or at least they're isolating them from any other genes' regulatory regions,
Mm-hmm.
right? Because there's a good diagram, right? Let's say these two genes were very close to each other, close enough that the enhancers for one, right, those could bend over and touch the promoters for another one. But you really don't want these two things being expressed at the same time in the same place. They're very specific to different tissues. And so what you have to do is you have to put a wall or a bookend between those two genes so that whatever's going on with this gene has absolutely nothing to do with this gene, right? And that's what insulators do. They basically silo bits of DNA. So this bit of DNA only, anything bound to this bit of DNA only affects this gene. It doesn't affect that one.
Oh, okay.
And that's what insulators are about. And as far as I'm aware, and I'm not an expert on these, sorry, cat fur floating around. Um, these prevent the bending of DNA. They actually kind of physically kind of like clamp onto it, like if you had a piece of wire, right? And then if you just clamp a tube around it, that piece of wire can no longer bend, right? Or at least not as easily. Or maybe it can bend, but it can't reach, like bend round on itself. And essentially, that's what the proteins bound to insulators. That's what their job is to prevent that bending. I'm pretty sure. So it's not really about preventing things from binding to DNA, which is what silencers are all about, or transcription factors that reduce gene expression. Um, it's more about preventing the topography of the DNA, the actual shape of it, from changing.
Oh, okay. That makes sense.
Yeah. Because these insulators could be in the middle of, kind of in DNA terms, in the middle of nowhere, right? They could be, you know, um, a long way away from any other regulatory region, let's say. But because they're able to stop this bit of DNA folding around over on the top, it stops this enhancer, or any proteins rather, bound to this enhancer from interacting with any proteins bound to the promoter on the neighboring gene. Right, because DNA can loop, and these loops can be, they can be millions of bases long. Right, these enhancer regions don't have to be close to that gene. right? So it's a very complex kind of interaction, and obviously the effect of insulators varies on how close they are to those enhancers that you don't want affecting other genes, right? Because if they're a long way away, then it's not going to have any difference, any effect. So like with everything that we've studied, it's complex, but that's the basic idea behind it. Okay, any other questions? We got time. It's going to be a short one today.
I had a question on slide 5 about the RNA silencing.
Oh, yay.
And I was just wondering,
My favorite topic.
what causes the transcription of both strands to happen?
Typically, it's that's a good question. So it depends. It's not so much that, well, I'm kind of trying to think, because this is something that I just take for granted, right? Because it's just a technique that I use. I don't tend to think about it an awful lot. And I've never actually been asked that specific question, so that's an interesting question. So one approach is that you have two opposing promoters very close together.
Okay.
So I'm trying to think. So one, yeah, okay. So I'm just kind of trying to gather my thoughts. So one is you could have two promoters very close together, so they'll transcribe separately, right? Small bits of RNA, and that RNA after transcription will then base pair. Another way is that this is a palindrome, right? So it's a sequence of DNA that reads the opposite, so that if you put a mirror down the middle, you'll essentially read the complementary DNA sequence for the other part. If that makes sense? I should probably try to draw this out. Well, we'll see how we go. If it doesn't make any sense, then I'll draw it out. And so then essentially you have like that, right? Two bits of sequence pointing in the opposite direction that when they're transcribed will be able to fold over and base pair, because they're the opposite of each other. But because they're pointing in opposite directions, it means that they can be transcribed in one go, and then they'll fold back round and they'll pair up. That's how the stem loop deal works. I'll have to have I'll read into the double-stranded RNA bit, because the only way that I've come across before is that stem loop formation where you have the palindrome. But for I'll have to check. Right? It's actually funny, because this is just such an assumed thing. It's like getting up and walking, right? You just assume that your legs are going to work, right? You don't think too much about it. And this is a technique that I've used for like 11 years. So I haven't really thought an awful lot about it, to be honest. The stem loop is I'm very familiar with, and I think in C. elegans, again, I'll have to check, in the RNAi constructs that I use, they actually use opposing promoters. With one single piece of the gene in between them, so that those opposing promoters will transcribe complementary bits that will then later on base pair. But I have to check. I'll check that up after between now and next class and report back.
Okay.
But yeah, something like either along the line of palindromes, right? So you have two bits of sequence which are in opposite directions but are complementary to each other. So when they're transcribed, they basically fold back over and form a double-stranded RNA like so. And if that's the case, if that's one continuous bit of RNA, essentially that loop at the bottom or at the top in the diagram on here,
Mm-hmm.
that then gets cut off, and then you just end up left with double-stranded RNA. Starting with double-stranded RNA, I think that would have to come from two separate promoters or two separate transcripts, but I'll double check
Okay.
so I can get back to you. But either way, the idea is that one of these strands, whichever one, so one of the, just going to look down here, right? When you're down into the RISC complex, you want to have a piece of RNA in the RISC complex, which is a bit of a tautology, right? Because it's an RNA-induced silencing complex.
Mm-hmm.
The piece of RNA that you want left in there has to be complementary to its target. So when this double-stranded RNA is produced, one strand will be exactly the same as its target mRNA or section of its target mRNA. And remember, these are very small. So mRNAs can be thousands of bases long. And these double-stranded RNA, probes, essentially, they're going to be complementary to a small part of that mRNA, typically to the 3' end, like the 3' UTR that we talked about before.
Mm-hmm.
So if you have a sequence, I don't know, AUCC, AUCC, let's say, right?
Mm-hmm.
Then in this double-stranded RNA, one strand would be AUCC, AUCC. The other one would be UAGG, UAGG, right? It would be the complementary.
Mm-hmm.
And that complementary strand, the one which matches the target, the UAGG, UAGG, that will be the piece that's left in the RISC complex. And that's the barcode. So you have the long mRNA kind of floating around, and the RISC complex kind of comes up, and it recognizes that one specific part of it, and then it cuts it. Or just stays bound to it and prevents translation. It depends on the degree of similarity. So essentially I don't know about all, most mRNAs in their 3' UTR contain those target sequences which are used to degrade them when these small non-coding RNAs are expressed.
Oh, okay.
And it's actually used a lot in, the best study system is in C. elegans, obviously, because it's the best model organism. But it's heavily involved in what's called heterochronic gene expression, which is gene expression that changes over time. And so in worms, they have a periodic molt when they shed their cuticle and they get bigger, like insects do, right?
Mm-hmm.
Or snakes. And so these microRNAs are really important in that timing. So essentially for a given developmental gene, you'll have this kind of waveform pattern of expression or production of the protein. And so what the microRNA does is it forces that downslope, right? It gets rid of that expression at a certain point in time. Right, so essentially the mRNA for a protein contains the information for its own destruction.
Oh.
Which, if you think about it in a bigger picture, makes a lot of sense.
Mm-hmm.
Yeah, it's super cool. We'll actually come back to that this class. It's one of my difficult things to talk about in genetics. Did that help?
Yes, that's it.
Any other questions?
No, that was it for right now.
Cool. Well, if you do have any, just pipe up. Okay, so, and also we have a recap session next Tuesday. before the class, the exam on Thursday. So what I'd suggest is spending the next few days going back through all the material since last exam and just put them together a pile of questions. You know, when you go back through it, there will almost certainly be stuff that doesn't make as much sense now as you thought it did back then. So make a list of those questions and bring them with you. I've also just read the PowerPoint slide. Actually tells you the double-stranded RNA is from transcription of both strands. There you go. I don't know why I didn't see that. There's a big arrow pointing to it. The stem loop is from the inverted repeat. Feel a little bit silly now. Anyway. Okay, so really this is just a, I don't know, kind of capping off the semester in many senses, right? This is a little bit of a look into some of both the applied aspects of genetics and also in many ways how we know what we know about the genetics that you've learned. You know, because the information that I've been sharing and the concepts and principles that we've been learning about are they're not inherently known, right? Someone had to discover them and figure out how different things work and how they tie together. And really a lot of that has been done through both mutagenesis, so forward genetics, which we'll talk about in a second, and reverse genetics and transgenics, right? So, and this isn't something that's stopped. It's going on, you know, as we speak, at least probably more slowly now that the everything's shut down. But this is a bit of a kind of window essentially into how you can study these things, right? And it's not really about making pretty glowing green rabbits, right? Even though supposedly they're very popular with children. Who would guess, right? But this is a cool slide because actually, let's just see if we can find something. Just do a bit of fun. Here we go. I should have just tons and tons and tons of pictures of this stuff. But essentially, this is a fave topic of mine because I use it so extensively, actually. So I'm being a little bit self-indulgent here. Let's see, what I'm going to look for is, well, this is actually a funny look through my organization. Here we go. Let's see. Yeah, that's kind of pretty. Fun enough, this isn't actually in indigenous fluorophores. This is in endogenous autofluorescence. So let's see if this comes up. Yeah, that'll be fine. It's an absolutely mahoosive picture. So this is an image of a fireworm. Right, this is a small invertebrate you can find in warm waters. They're called fireworms because they have spines on them, which will cause an intense burning sensation in your fingers if they get through your skin. I'm just trying to find some. Here we go. Isn't that beautiful? So these are like the spines. I forgot the exact name of the structures. It's not what I work on. This is what a colleague of mine works on. But this was taken using a confocal microscope to basically look very precisely at fluorescence. Let's see. Let's see if I can get. I'm just being a little bit self-indulgent here, to be honest, Rose. So let's see what these look like. Yeah, these aren't especially. Oh, this is actually a fairly decent picture, right? So this is a worm, right? You wouldn't know it, right, from looking at the picture. And this is a fluorescent marker for a cell at the very, very tip of its gonad. So this is where germ cells are produced, and you can actually see it has like this weird kind of mesh-like structure, right? And that's what's called a germ cell niche. And so you can actually look at it in 3 dimensions. I don't have the models open, but you can actually rotate these around kind of in all kinds of orthogonic directions. And here you can actually see these blank spaces are germ cells, right? And they're actually produced by this cell here. This is called the germ cell niche, right? And so the reason why this glows is that this worm has a construct, right? A piece of DNA made, not necessarily artificially, but it's not endogenous to the worm. It's not naturally found in it, let's say. And it is a piece of regulatory sequence from a gene called LAG2, right? Which is why it's LAG2 is in the file name. And that gene is expressed in this one cell, or 2 in this case, right? Because there's 2 different gonad arms. They go like so, like horns on a bull, and they kind of overlap slightly, right? Which is why you have this kind of 3D nature of this image, right? They're not in one single plane. They're in a slightly different plane. And then you put that promoter region, right? That regulatory region which controls expression of whatever's downstream of it, and you put that in front of a green fluorescent protein, which is from, let's see if I can spell it right. I don't think that's right. Yeah, totally didn't spell it right. I don't want Victoria's Secret. That's not what I'm interested in at all. And so essentially it's a jellyfish, and that jellyfish glows just naturally. Let's see if I can get some. There you go. Isn't that pretty? Got there eventually. So this jellyfish uses a protein which fluoresces. So essentially what it does is it produces a burst of blue light, I think by calcium release. And that blue light, which you can see here, then excites this protein, much the same as light excites chlorophyll, for example, in chloroplast during photosynthesis, or the blue light in the LEDs or fluorescent tubes above your head excites a phosphor, which then re-emits a different color. And so this protein, and you can see a look of it here, it's a beta pleated sheet barrel, basically, absorbs that blue photon and then kind of re-emits that as green. And so these jellyfish glow green. And there are lots and lots of other different colors as well. Corals are another source of fluorescent proteins as well. And you can get red, you can get cyan, you can get yellow, you can get green. Those are the typical colors that you would use. And so this isn't really about making stuff look pretty. It's very much about using this as a tool to look at function, right? So, and actually, do I have my paper open? Yeah, I do. How about that? Forgot to close it. This is a paper I wrote a few years ago. So this is, I think I talked about it in the audio overview about a gene with multiple promoters, right? So this is a gene called PNC1, and you can see this is the head of the worm, right? So this is where its brain is, essentially in its mouth. And you can see that this protein normally is expressed in all kinds of different places, right? It's expressed in neurons, it's expressed in muscle, these are intestinal cells. Morning, love. This kind of diffuse fluorescence here shows that it's extracellular, right? So that's proof that this protein is secreted, right? And so you can ask an awful lot of really, really interesting questions using just this technique, right? It's super, super cool. And it's one of the RNAi and fluorescent proteins are the two main techniques that I use in my lab to do my research. And one of my projects, actually, which is kind of finished, sort of, is using a fluorescent protein as a reporter to see how active a protein is. Is it retained in the cytoplasm and inactive? It's a transcription factor. Or is it activated and goes into the nucleus? So you can actually just look at a worm and you can see the ratio of cytoplasmic to nuclear GFP, and you can infer from that how active that gene is. So this is a super, super, super cool technique, and there's lots of... Hey peeps, don't forget that I'm on a class right now, okay? It's a super cool technique, but it's just one of many tools that you can use to look at gene function, right? And so this is really kind of a window into how you can do that in, genetics. So in terms of language, right, there's really two, when you're looking at gene function, right, or trying to figure out how something works, let's say, right? How does limb development work, for example, or what's the how does immune response to bacteria work in worms? You have two kind of general approaches. One is what's called forward genetics, and this is very much outcome blind. They're commonly called fishing expeditions because we don't know what you're going to catch. And essentially that's through mutagenesis, right? You kind of mutagenize these worms, organisms, right? Not everybody works on worms. And then you look to see, do I get a phenotype that I'm interested in, right? Has the vulva disappeared? Do these worms die from bacterial infection, whereas normally they would survive, for example? And then you work backwards from that phenotype to try and figure out the genotype. Okay, where is that mutation? What's it in? What change does it produce in that gene and its function? The flip side of that, so that's great for finding out stuff that you don't know, right? And so if you've got no idea how this thing works, a forward screen is a good way to start, right? To kind of start looking, essentially. Now, reverse genetics is the opposite. Essentially, okay, I know that this gene, let's say alcohol dehydrogenase or lectin, right? I'm pretty sure that this is involved in a particular process. Now, what I want to do is I want to work backwards from that to its function and try and figure out how this thing works, right? And so that's reverse genetics. Right? That's very much starting with what you know and then working back to something that you don't know. Right, and that's very much more kind of modern recombinant transgenic type work, like RNAi, for example. So I'm not going to talk a whole bunch about this because this is really kind of like a central principle of this work. If you want to make any of these changes, you have to make them in a way that they're heritable, right? So essentially you want to, if you're going to modify an organism, you've got to modify it so that that modification is present in its germline. And so by doing so, that modification gets passed to its offspring. That's really all that's about. So there's no point mutagenizing an organism and that gene of interest, the mutation that you're interested in, being its somatic tissue and not in its germ tissue. Because as soon as that organism dies, that mutation dies with it, right? And that's also why, in a sense, you know, it's like if you get skin cancer through UV-induced mutation, that doesn't get passed to your children, right? Because it's in your skin tissue, it's not in your germ tissue. So there's a couple of different approaches, right? There's induction of mutations, right? Specific ones, and these are called knockout mutations. And there's gene knockdown, right? So there's two different ways of looking at this. If you knock out a gene, it doesn't work, right? However, and that's really powerful, right? Because if you have a particular gene of interest and you go, okay, I want to study what does this do, you could either do it by forward genetics and just try and hope that a mutation that you're inducing occurs in that gene, right? Which is an enormous amount of work, and it's no guarantee of success, essentially. Or you could go, okay, I know what gene I want to work on. I'm going to deliberately mess it up so I can study what effect that has on function and you know, broader processes. Now, the flip side of that is, I'm going to ask you a question, Rose. Make sure you're still there. What would happen if you did a knockout mutation, let's say in one of the ribosomal proteins? Think about that. What would be the consequence on that organism and its offspring if you knocked out a ribosomal protein?
Would that mean that it wouldn't necessarily, the genetic information of that organism or offspring wouldn't be translated because there's no ribosome, or the ribosome doesn't work?
That's correct.
Okay.
So what broader consequences on that organism do you think that mutation would have?
It may possibly die, or I don't know.
Right, exactly. So if you had that mutation and you're homozygous recessive for it, would your offspring ever make it to adulthood to be able to be studied, let's say? It'd be a no, right?
Yeah, no.
Even if you were somehow able to survive, your offspring wouldn't. So there are a set of genes in any organism which are essential. And if you knock them out, you'll not get a viable organism. Oh, one of our cats meowing. Those are called lethal genes, right? So there's a whole category in C. elegans, at least 1,000 of them by now, that are called LET genes, short for lethal. If you knock them out, the worm dies, right? And so they're very, very hard to study through this particular approach, or even just through random mutagenesis through forward screens, even though a lot of them were characterized that way. You know, that's how we know about them. It's really, really hard to study them because, you know, if they're homozygous recessive, no worms. So the alternative way of doing that, or at least kind of the old school alternative before CRISPR-Cas came along, is you can do gene knockdown. And so what that's about is you're not changing the underlying genetic information of the organism. Now, if you want to study ribosome function, let's say, or even ribosome function in particular tissue or a particular time, now you can knock down, right? You're not knocking it out. You're just reducing the level of gene expression to the point where that protein doesn't function, right? It's not present. Now you can study those lethal genes without worrying so much about getting offspring. Obviously, it's still going to be difficult to do. And I've done a lot of these in the past where you end up getting a very small number of offspring, but you're still able to study the effects of that, right? And so gene knockdown on, and in C. elegans, this is called RNAi, which stands for RNA interference. This is essentially using this technique, right? So remember we talked about this at the beginning of the class. Essentially what you can do now is you can create a construct which has a small piece of target mRNA. Let's say you want to knock down ribosomal subunit. So now you clone in a small piece of that gene into this construct, either between these two promoters or as a kind of a palindromic repeat or an inverted repeat. And now you express that in, you know, your tissue of interest, or in the case of worms, you just feed them bacteria that are producing this double-stranded RNA, and that will then reduce the amount of mRNA produced from that gene to the point where you don't get function. And so then you can actually start looking at, okay, what role does this particular ribosomal subunit have in translation, for example? So there's two kind of very complementary approaches, essentially. Does that make sense?
Yes, I think it does.
Cool. Okay. Well, if it doesn't, you know, just bring questions. That was the wrong one. Next class. And so there are a bunch of different ways in which you can create these knockouts, right? And essentially, this is kind of old school way of doing it, right? But you can do some real cool stuff. So you can generate obese mice, for example, which seems a little cruel, but they are super cute. They're like spherical mice, basically. And this is the result of knocking out a hormone called leptin, not lectin. That's something you put in jam, I think. Leptin, right, which is essentially a fullness hormone. Tells you to stop eating because you're full, right? Because you don't need to eat anymore. And so if you knock that out in mice, essentially those mice never feel full and they'll eat constantly and become fairly chunky. So the big deal in the last, I'm trying to remember when this actually came out. I think it's probably no more than 10 years ago, right? Which may seem a long time to you, but it's it's not a very long time in terms of science. Is something called CRISPR-Cas9, and so this is it's hard to understate how much this has revolutionized molecular biology and molecular genetics, right? Because this allows you to very precisely target your change. So essentially, you can read through all those things yourself. Basically, the idea behind it is you have a target, right? And this PAM sequence is a short set of nucleotides that just occurs randomly. You need it for some reason that I don't fully understand. But you have this target DNA, and you're going to design yourself as a scientist a guide RNA which is going to match that target DNA, right? It's by complementarity, right? So it's going to complement base pair to one strand of that bit of target DNA. In addition, and this is a third part of the deal, you have a protein called Cas9. And so Cas9 is both a helicase, right? So it unwinds DNA, which you can see here. And it's also a DNase, right? Endonuclease, actually. It cuts DNA, causes a blunt end double-stranded break. And so essentially what you want to do is you want, okay, I've got this target DNA that I want to disrupt in some way or insert something into. You design a guide RNA to match that. And so when you inject that or get it somehow into your cells of interest or organism of interest, you also introduce protein. And so what happens is that guide RNA and the protein kind of assemble, form this what's called an effector complex. And that effector complex scans the genome essentially, right? And it just kind of wanders around until it finds the target DNA using this guide RNA as a way of matching that. And that's very similar to how the RISC complex uses its bit of single-stranded RNA that is complementary to its target RNA. So it's a very similar concept. Let's get back to this one. Now, when it does bind, what happens is the effector complex unwinds that double helix and cuts it. And obviously that's bad. And so typically what happens when you have a double-stranded break is the cellular machinery just goes, ah, and starts running around trying to fix it because it's pretty catastrophic. And the typical way in a non-dividing cell would be oh, well, crap, we just got to stick this back together kind of response. And that's called non-homologous end joining. And so simply you'll get proteins which recognize a flapping around DNA end, right, which doesn't have anything on it because those shouldn't exist, right? Because you have telomeres typically at the ends of chromosomes. And it will find another flapping around N and it will just stick them together. All right, it's very much what I call the Hail Mary approach to DNA repair. It's like, even if we might get this wrong, it's better than not doing it at all kind of thing. However, in the process, because this is not a very good way of fixing DNA breaks, particularly double-stranded breaks, typically you'll introduce either an insertion of a very small number of bases or a deletion, right? So it's not a very faithful fix. You end up modifying that bit of DNA where that break was. And so if you target your guide RNA to, let's say, the middle of an exon, exon 1 of a protein, of a gene rather, then that duplication or deletion is almost certainly going to introduce a frameshift mutation, and it's going to stop the production of that protein, right? So this is the mutation that's introduced is random, right? Because you don't know what it's going to be. But the place in which you introduce it is not random. That's very deliberate, based on where you design your guide RNA to recognize. Does that make sense?
Yes, I think it does.
Okay. Yeah, so as a scientist, you choose this guide RNA. Actually, you probably just put your sequence into a program and it will tell you what the best ones are. That's what I would do. But you choose out of those which ones you want to use. And you probably do a whole bunch of them, see which ones work best. Now, the other thing, so that's in and of itself, this non-homologous end joining induced mutation is amazing, right? Until this came along, you could not do a targeted mutation in C. elegans. It wasn't possible. You could in other organisms, sort of. It was kind of clunky, but in C. elegans, if you wanted to knock out this particular gene, you just had to do it through forward genetics, mutagenesis, or hope someone else had done it, right? And so it was very frustrating, right? Particularly if you wanted to knock out a particular part of a gene and not another part, let's say. So that in and of itself was amazing. Now it gets better. And this is really where CRISPR-Cas9 has just changed everything. It's really hard to overstate this, because when I first started going to worm meetings, there's a international C. elegans meeting that's held in California every 2 years, or at least was. It now alternates with a European country. When I first started going to that in 2011 or so, people were like, oh, there's this amazing new technique this is going to revolutionize everything. And then after that, every 2 years, more and more people are using it to look at more and more things in all kinds of different ways, and just basically discovering things that it was not possible to discover before, simply because of this one technique. It's just astonishing. And actually, to show you how important this is, there's been a multi-million dollar, probably even multi-billion dollar by now, legal battle between two groups which claim to have discovered this and patent it, because that's how important this is in terms of money. Lawyers are making out like bandits, that's for sure. Anyway, so the thing that really changed everything, even more than just this ability to do targeted mutations, is if when you induce a double-stranded break, there is a matching piece of DNA around. So this might be a sister chromatid during cell division, right? This is a typical way it works. But if you provide that, now we use a different method to repair that break. This is now homologous recombination. And essentially what you do is you have your, did we talk about this in terms of DNA repair? I really can't remember whether we talked about it in genetics or in my other class. I think we did, I'm not sure though. Can you remember?
I'm not sure. I know we did talk about like the holiday junctions, but
right. Yeah, so we did talk about the repair of double-stranded breaks then. Yeah, we did. Okay, cool. I know because I talked about it in my other class as well. I couldn't remember if I'd done it in this one, but we have. So essentially, normally in double-stranded break repair or homologous recombination in prophase, you have another matching template, right? Exactly the same. And so if the bottom one is broken in two, and you have an overlapping one on top, now you have proteins that recognize those two bits and those two bits and say, okay, these go together, and then we kind of fix it through the mechanism that we talked about in class, homologous recombination. Now what you can do is you can essentially trick the cell into using a bit of DNA that you provide, right? So if you provide a piece of DNA which has matching ends, right, which is going to match either side of your targeted break, you can put whatever you want between those two ends, and that will get incorporated into your target site. So let's say you want to change one nucleotide in that gene to change one amino acid, let's say, or introduce a stop codon or whatever, right? Now you can provide a piece of donor DNA which has an exact copy of that gene with that one change in it. Now when that DNA double-stranded break is fixed, that copy is inserted into that break in order to fix it, right? And so now you've made a not just a targeted mutation, but a deliberate known mutation of your desire, right? So that obviously expands things even further. You can also insert, if we go back up here, right, this is a fluorescent worm, and almost certainly this was as a result of a really old school clunky way of getting a construct into worms. You inject them, which is I've done before. That's how I generate those other ones. But that DNA doesn't become permanent, right? It's like a plasmid in bacteria. If you want it to be permanent, essentially what you do is you drop those worms into a nuclear reactor and just screw them up, and you form a whole bunch of double-stranded breaks, and those kind of get fixed, by non-homologous end joining, and your bit of DNA might get incorporated into one of those fixes, right? It's a super clunky way of doing it and doesn't work very well. When I tried it, I didn't get what I wanted, unfortunately. It's cool to go to a nuclear reactor though. Now what you can do is you can have that construct, that DNA encoding a promoter and a GFP reporter, right, green fluorescent protein reporter. And if you put homologous ends, let's say you don't want to screw up a gene, you just want to put this into somewhere innocuous, right? So it doesn't disrupt a gene's function. Now you can provide your donor DNA is that reporter construct, that promoter and green fluorescent protein gene, and that will insert it into your organism's genome. And so now you have a stable transgenic, right? Every single offspring of that worm is going to have that construct. Every single cell is going to have that construct. Whether it's expressed or not depends on the promoter. But now, instead of just doing it like a real old school clunky way, you can do it in a very deliberate way too. And you can control dosage, right? You know, so doing it this way, you've got no idea how many copies of that reporter get incorporated. It's just totally random. Could be 10, could be 100. So different strains of worms, you know, glow brighter or dimmer or in different places. Whereas now you can be very, very deliberate about it. So it's kind of hard to overstate how groundbreaking this is. And also, if you listen to the audio walkthrough, I don't know if you had a chance to or not, but talk about a case where this has been used to edit human genomes. Two girls in China have had their genomes as embryos essentially modified using CRISPR-Cas9. So it's very much kind of getting into the genetically modified humans range, which is a little disconcerting. But now we have the power to do that. Obviously, it doesn't mean that we have the right to do it, or if we would be legally or ethically allowed to do it, which we certainly wouldn't, which is partly why it happened in China and not in, say, the US or the UK. But that doesn't mean we can't do it. We can. So just think about that. That's a bit of a Pandora's box, I guess. So yeah, that's pretty much all I have. Any questions?
I don't have any right now.
Okay. Well, have a think about this stuff. It's fairly straightforward, but there's an awful lot of stuff going on here. But yeah, just spend the weekend going through the material from the last few weeks and bring everything you got with you to class on Tuesday, and then we'll just go through them one by one.
Okay, sounds good.
Awesome. All right. Have a great weekend, Rose.
You too, sir.
Yeah.